Our next speaker is Dr. Markus Covert, Associate Professor and Director of the Allen Discovery Center for Systems Modeling of Infection at Stanford. Markus?
Thanks. First of all, that was great. I loved it. And it's amazing to see how quickly that institute has taken off, and very excited about those resources. Of course, also so just really thoughtful about Paul. That was really surprising and hard. Like Rick said, you know, he's touched all of our lives, but it's amazing to see just how many lives he has touched in so many ways. And so really grateful for him and thinking about him. And, you know, excited to talk about what we've been doing kind of with his, I'd say, visionary support. So I'm going to talk a little bit about the center as a whole. I was kind of encouraged to do this at our site visit, and I'm going to use a lot of the slides from our site visit. So it's going to have kind of a center feel to it, in addition to hopefully some fun science. So the key thing that our center is dedicated to doing is trying to open new windows into infection. Okay, and in our case, we're particularly considering macrophage and interactions with Salmonella and E. coli. And we're hopeful that by bringing to bear cutting-edge modeling, computational, and experimental tools in like one big integrated approach, that we'll be able to shed light in ways that really haven't been possible before. And what we've done, we've taken kind of these three circles of modeling, computation, and experimentation, and we have tried to get after these things, first of all, by trying to really push large-scale cell modeling forward. And when I look at what needs to be done to take a large-scale modeling to the next step, where it can be used for real kind of large-scale applications, I see two major axes. One is that we need to go from simple bacteria to more complex, increasingly more complex cells. And the second thing is that we need to be able to go from modeling one cell to modeling large groups of interacting cells, right? So that's the way that we can go to tissue, to more complicated interactions, and hopefully eventually to whole, large, complicated organisms. In our case, what we're trying to do is get directly towards how pathogens interact with their hosts, their cell hosts. We're also trying to come up with new computational tools. You heard some great stuff just a minute ago, and we're definitely right in that space as well, trying to do rapid analysis and also visualization of both the biological and modeling data. And then finally, we're developing new experiments, right? We've found that in a lot of cases, the reasons why we can't model what we want to is because we don't have the data that we would need to have, and it's because people aren't able to generate that data, so we've invented some of those technologies ourselves. We have a great team. What I love about the team is that it's truly very diverse experience. Denise and Jerry are here. I hope you've had a chance to talk with them, and I'll be talking about some of their things. The group has gotten pretty big. We're now like 50 plus people, and it's been really exciting to see what's been happening with that. This just shows some of our kind of touch in these different areas. There are two things that I want to mention in particular that really the Allen Foundation enabled that I would just push to you programmatically because it's very interesting to me. One is that we were able to bring on someone like Jerry, who is right here, who is an amazing, historical software engineer. I don't know if you had the chance to talk with him at the Computer Museum yesterday, but was one of the team who made cut and paste, which some of you are probably using right now. Also put maps on the first iPhone, right? An amazing person, but not a traditional PI, right, in the NIH sense. But here, thanks to this grant, we're able to bring him on as like a full, PI who is able to manage a software team. And that has been transformational for us. The other thing that's really transformational is we have a concept of strategic commissioned work, which basically means that we can very rapidly and flexibly see if somebody has a brand new technology that would be of obvious interest generating data for us or generating new tools for us. We can flexibly go and just offer to commission some work from them that enables us to get things done that otherwise wouldn't be. So that's been huge for us, just on the programmatic side. These approaches are very much multi-scale, and I'll just show this really briefly, but I want you to get a sense. Like, the modeling is occurring, as you see here with kind of these specific projects, from the level of modeling proteins interacting with each other to all the way to host pathogen modeling. Those are very different strategies, but they're all together. And in computation, it's the same way, right? We go from working on software that will benefit individual labs in the center to whole center. And we even get out, as we distribute our materials and as we use crowdsourcing to help us generate the training data that we need for some of our sets, we really are looking, again, from individual labs to like the global community, including non-scientists. And finally, at the experimentation level, we're making both kind of time-dependent single-cell resolution measurements in bacteria all the way up to what I'm very excited about, thanks to Denise, really, getting us into these in vivo studies where we're able to really look at a whole animal and its interaction with bacteria, et cetera. OK, so let's talk a little bit about what's going on in modeling. As some of you know, we've been working towards, we started with this whole cell model of Mycoplasma genitalium, and we've been pushing this to E. coli, taking all of the information that we can possibly take. This is now hundreds or even thousands of papers that we've pulled together, millions of data points, and trying to represent, and very heterogeneous, which is what this is supposed to say, and trying to represent all of that in one integrated model. And the way that you do that is by taking, like for example, in this case, we can write 4,500 equations which correlate to the, which describe the RNA in the cell, right? Another several thousand that describe the metabolites, then a whole another set of 4,500 that describe the proteins in the cell, right? In that way, we can explicitly encapsulate all of those data and bring it together in one integrated form. We then take those equations and that data and we pull it into different functional categories. And in that way, through an integrated process, which I don't have time to describe here, we're able to represent cells, right? Cell behavior. In the case of E. coli, we don't have a whole cell model yet. We have what we're calling our half cell model. But we're well on our way to being able to explicitly represent by a clear majority of the genes in the cell and their known functions. So just to give you a sense, like in this particular case, we were able to identify millions of data points generated over the last century in E. coli. And it's kind of amazing, right? Because in some of these cases, you have a data point that someone spent their entire PhD basically trying to generate, right? 1 point. In other cases, with these new high throughput studies, you have people who've generated, millions of data points in a week, right? But very rarely are these things done in true coordination where people are thinking, what's the ramification of this data point with all of the rest? But we're able to do that by taking these things, as I said, bringing them together in equations. In our case, all of this curation led to the identification of 19,000 parameters from the literature, which are then encapsulated mathematically. So those color bars are to tell you, if you're able to see the whole figure, what how those data slot right into this thing. And then we build that into this unified model that can take all of those things and we can say, okay, how self-coherent is this data? Like we can cross-compare it. We can also see how well it's able to make predictions about cellular behavior. As you see here, we can run simulations over many generations that have all of this capacity to just have so many windows into the cell. So that's been really exciting. And I'll show you an example of a simulation here. Here you see E. coli. It's growing in a minimal medium condition, actually. You can see the DNA being replicated over here. And one of our things we're really excited about, the original whole cell model could only do 1 generation. Here we can actually do kind of an arbitrary number of generations because of new mechanism that we've incorporated into this E. coli model. So you can see as it grows out and replicates. Now what's going to happen in a minute here is that it's going to now have an environment shift. Okay, we're going to move from our normal cell environment to an amino acid kind of containing rich environment, which is going to lead to a number of phenotypic changes. So if you look up here, let's wait 1 second. Yep, there's the nutrient-rich environment. This is, by the way, all based on real data. Everything behind this animation is real simulation data. So what will happen is a bunch of sensors will sense that the amino acids are now in the medium. That will change the expression of many genes, just a subset of which we're showing here. So you'll see circles getting bigger and smaller. Same thing with the concentration of metabolites. And as a result of all of that, you'll have this shift. You can see that now the cells are getting bigger. Okay, they're growing faster. And what's eventually going to happen here, right, so E. coli is going to start parallel processing, right, generating multiple chromosomes at once to be able to maintain that new faster growth rate. So pretty exciting for us to be able to capture a bunch of these new behaviors in these cell simulations. We've noted a number of things in here so far. The thing that I'm most excited about was kind of brought up in some ways by Jim yesterday, talking about antibiotic resistance. One of the things that we've found is that if you look at the limits, the volume of the cell, the time that it takes to grow and divide, it turns out that at least 50% of the gene content really over every environment is not expressed every cell cycle. So that's a really interesting observation. And one of the things that I found most interesting, if you look at the functional categories, like 80 or 90% of the antibiotic resistance and persistence genes are exactly in that category. They're not expressed every cell cycle. And so it gives you this idea of where persistence comes from and natural variability in an E. coli colony. What we're doing right now, we continue to push towards having a whole cell model of E. coli. We are also trying to incorporate some of these lessons that we've learned into making not a whole cell or even a half cell model of a macrophage, but something that can incorporate multiple functionalities using our framework. And what I'm super excited about right now with Shane and Jerry, we've been working on trying to model, again, I told you about these two axes, right? More complex cells and more cells interacting. This is literally just basically a week off the press, but we're now able to, for the first time, look at multiple cells interacting together. Just to give you an idea, when we did the first whole cell model, it took about 10.5 hours to run one cell dividing one time. Thanks to a lot of the software innovation, we can now do similar simulations in about 10-15 minutes on a laptop as opposed to on a cluster. So we've just made huge strides, and that's been enabling for us. If you look at this, I mean, I could barely look at this without geeking out. But okay, so he's clicking. This is Ryan, one of our software team. He's clicking on this screen. Every time he clicks, he starts a new instantiation of the whole cell model, which is or the half cell model, which is what you're seeing in the background. And to do that, you had to be able to run all of these things in parallel, interacting in real time with an environment. The environment is its own thing that has to be modeled, right? It has 80 components. And so what he's doing here is just showing you different components. As E. coli is taking up those resources, you can actually see what's happening to the environment. And diffusion is all included in here. So if you were to watch this over time, you'd see how it's backfilled as diffusion kind of replenishes and changes the overall concentration. So when a cell divides, a new instantiation is created and initialized. So this is what keeps me up at night right now. I love it. So that's very exciting. OK, moving to computation. And I can go through some of this kind of quickly because there definitely is some good overlap with some of the things you saw with the Cell Science Institute. So we are spending a lot of our time trying to fight the dirty secret in live cell imaging, which is just that it is so hard to analyze images, especially live cell, especially, I'm sure, even more with 3D. But we've come up with a suite of tools as well that are pretty flexible that you can use to, that anyone can use to analyze their cells. And so here's a set of IPython notebooks that we use. Maybe the more kind of well recognized at this point is the deep learning that we've been using. I'm showing you here for bacterial cells because it's a problem any type of cell you look at. But here is an example of a colony that even when it's this overgrown, it's almost impossible to be able to get what you want out of it, right, in terms of automated segmentation. But the deep learning just can do it easily. This is no curation at all. So that's pretty amazing. We can also do this with nuclei, as was said, with cytoplasms. It's amazing to see what you can do with even with no labels. We've also co-cultured different cell types together, been able to tell them apart. And that's on a tool called DeepCell that you can feel free to look at, and that now Dave, who really drove this, he's He's taken on a new independent professorship, but he continues to run some of this through one of those strategic commission work mechanisms. So we're using crowdsourcing to try to get all that training data that was spoken about. That still is a problem. There's overhead. It would be really great if there was a repository where everyone could deposit their training data, because then you would have these deep learning tools that could literally just look at anything, right? And I think it's only a matter of time before the NIH or somebody does that, where there's just a wide library of training data. This is what we're hoping to do with that, as you'll see from the experimentation, is to be able to look at mammalian cells and infecting bacteria, automatically recognize them all in the same images. We're engineering the E. coli model, as I already talked about. And another thing that we're doing a lot of is just thinking about how to interpret stuff, right? And one of the biggest problems, I think, is in this, one of my favorite XKCDs, right? This whole problem that correlation does not mean causation, right? So you look at these things and you think, okay, I've got some correlations, I've got some analysis, how am I actually going to get a mechanism out of this? Now, the bigger problem that most people don't talk about, but it's just as important, is that causation does not even imply correlation. Okay, so if you were to look, for example, one of my favorite examples of this was in Sunny Shi's lab, where they looked in a single cell and they just counted the number of RNAs, counted the number of proteins of the same kind. The causation of DNA to RNA to protein is maybe the most fundamental causation we have in biology, right? And yet if you look in these individual cells, there is no correlation between the number of RNAs and the number of proteins, and it's for a variety of reasons. Now, an interesting thing is, if I could sit down with anyone here and we could write a 20-line piece of code that would encode that causation, and not only would we be able to represent the actual results that you would see in this experiment, we would predict that there would be no correlation between RNA and protein in a number of cases. So getting at causation is really problematic, and correlation is really not the key, no matter which direction you're coming from, whether you're hoping that causation goes to correlation or you're hoping that correlation is going to get you to causation. So we've been thinking a lot about causality, and because we have this great whole cell network, we're able to do a visualization. This is a little stylized, coming from one of our strategic commission work partners, but we've made now a tool that goes through these simulations that we generate. And what's really neat is we have a causal network, a causal map of our network in the background, so you can look for anything that you're measuring or simulating. You can see what's upstream, you can see what's downstream. What's more exciting is that every node in that network is connected to the dynamic trace of the simulation that generated it, right? And so now you can do things like say, okay, I want to see everything in my simulation that has the same dynamics. Okay? Or you can try to trace back a pathway. If you know that there's a perturbation and something changed in the behavior you're observing, you can click back and back and back, and you can get to the root cause. This has been really amazing for us. We're not quite ready to go live with it yet. This is also one of these things that we just got a couple weeks ago. But I just want to give you an idea of what's to come. I think as we start looking in these very multifactorial complex ways, there are going to need to be new visualization tools, information design tools to get us to like a real understanding. How can we grapple with that complexity? And I think that this gets us to something really exciting, namely an ability to really systematically dissect causation. Okay, finally we go to the experiments. Some of these things I've talked about before, but they're a lot of fun for us. Like one thing, we've spent a couple years working on trying to figure out how to measure signaling dynamics, like let's say of some transcription factor activation in this case. Take signaling dynamics and in the exact same cell, after watching the dynamics of some behavior or set of behaviors, to be able to get the full global transcriptome by sequencing or by FISH. So that's what we're showing here using the Fluidigm C1 microfluidics platform. We're able to, in this same individual cell, look at the activity of, let's say, a signal transduction process and the downstream expression in the same cell. So that's been exciting and very important for modeling. Maybe what one of the things I'm most excited about that we're doing with Denise right now has been to be able to look again, in the same cells. These aren't all synchronized, unfortunately, but what you're looking at are macrophages. These are all the same. It's the same view. These are all macrophages that are expressing a new kind of kinase reporter that we developed called kinase translocation reporters. A reporter of transcription output, NF-kappa B, the growth of salmonella, right? And you can see all of those together in the same cell, so we can really try to look at these infections and see what they're doing in real time. The most exciting thing to come out of that, I could show you, the graphs are pretty complicated, but the really exciting thing for me is to see that it turns out that these macrophages can really tell the difference between a wide variety of situations. So they can tell the difference between if they just have bacterial cell debris around them or real bacteria inside them. They can tell if they are infected or if their neighbors are infected. They can tell if, like in this case, right, so they can, if there's just salmonella around them versus inside them. They can tell if they have a pathogen versus a non-pathogen. And they can even tell if the bacteria inside them are actively growing or if they're just kind of dormant, right? All of those things are, you can clearly see them in the signals that are being sent, which to me is just astonishing how nuanced and specific these cells are able to be in terms of response to an infection. To me, maybe the most surprising and exciting thing is, we are several years early, thanks again to Denise and Casey's hard work, getting into animals. And so one of the things they've been able to do is look at the microbiome. I won't do this justice, so definitely make sure you ask Denise about it. But the key insight here was that Bacteroides, which is another bacterial species, right, and Salmonella seem to interact together in ways that that can be dangerous or not. And a key mediator, which now looks like it could be a really interesting potential for a therapeutic, is this intermediate propionate. So very excited to see where that study goes and kind of ongoing work. Moving forward with these, we really have just a few exciting things. We want to follow up on some of these things that I've already shown, really is what I want to talk about there. The last thing that I'll mention is that we've spent a lot of time trying to make these things available to everybody, and it's been great to see how it's been accessed. You know, from that deep learning work, which is now in less than 2 years, has been clearly exciting. As we saw yesterday, you know, AI is of great interest these days, but it truly is so powerful, as I hope you saw also from Rick's stuff, in terms of analyzing images. And so a number of other distributions. The last one I guess I'll mention is just these kinase reporters, which are which I've told some of you have asked about over the last couple days. So with that, I just want to again thank the amazing people that we have in our center. It's been really great working with them, and really just astonishing how much we've been able to get done. And then finally, of course, like I'm sure so many of you, you know, Paul has really changed my life and changed all of our careers, and it's just kind of amazing to think about what he's done. And I loved what Alan said yesterday, right? The best way to honor him in some ways is just to get out there and kick some butt with science, and I hope we're all able to do that. And with that, thank you very much.
We have time for some questions. Jeff?
So one thing, going back to the 19,000 data points, and some from an individual PhD and some from, how sharp are each of those data points? And I'll explain what I mean. Are these simplifications that many people as humans want to do out of the causality correlation, or are they distributions with fuzziness in each one? And this sort of also ties in with what Bassam was talking with us about yesterday. Does that make sense?
It does make sense.
Because that would change things sort of falling off cliffs combinatorially.
Right. The data is, as you can imagine, crazy and hairy, right? Yeah. So the 19,000 is really our distillation of many more data points into what we feel like is, this is a trustworthy, somewhat maybe self-consistent set, right, of things. There's no fitting in those parameters. It's all just trying to take from the literature. Now, I'll give you some interesting ideas.
But the real question, sort of the follow-up, is when you place that into X thousand equations,
yes.
Are those, to use the overused, are those deterministic equations or are those fuzzy logic equations?
Yeah. It depends on, so I'll give you kind of a simple answer to that, and then I want to go to a couple things you alluded to in the data that I think will be surprising to people here. So the equations that I show here, right, we don't use a set of pure ODEs, right? So the reason that whole cell modeling works is because it's kind of a patchwork of many different types of models, which exactly like you say, some of them have probabilistic components, some of them are strict deterministic. So it's highly variable depending on what we judge from, building any kind of model is going to be subjective. But you can imagine in some cases, what we have are detailed single cell level resolution dynamic measurements that we can get those kinds of parameters out of. In other cases, there's a figure 7 in cell with an X that has an arrow with a question mark on top of it pointing to a Y, right? So there's a wide variety, and we try to never over model what we have, if that makes sense. Going to the data, I think it's very interesting. So we first noticed that this would be an interesting issue with the original whole cell model, and I'll give you the simplest example. So we were interested in what the mass is of DNA, right? That's an important parameter of our models, right? If you go in and measure, so it turns out somebody had measured that in the 50s, right? They'd done it the way you would expect, kind of dried cells out, cracked them open, measure. I don't want to oversimplify. But now, thanks to the genome sequence, right, we actually have another insight into what the mass of the DNA is, because we can just add up, take the sequence, add up all the molecular weights, right, and we get. So it turns out if you do that, the measurements that they made in the 50s aren't even enough to account for 1/5 of a chromosome, right? So this is a clear case where these data are totally inconsistent, but you wouldn't have necessarily, I don't think people have really been cross-checking them against each other, right? It gets much more complicated. And like you said, sometimes it's trivial, right? Like you go to the kinetic parameter measurements, somebody measures a 1.9 for some km, let's say, right? Weirdly enough, right, there are maybe 3 or 4 papers that happen afterwards, and they all are 1.9, right? So that's a problem. It's actually, you'd like to have more distribution, right, in that. There are also other cases where you take the measurements that have been made, and they're not enough, like I talked about before, to account for a cell or to account for a growth rate, right? So those things are very interesting, and that's kind of been the first major paper that we have written based on this stuff is taking what we found to be the 4 or 5 major discrepancies between those data and trying to resolve them. But I think you can't do this without a model like we have, right? That's the only way.
Okay, thank you so much, Markus.